Good morning, and please take your seats. We've been charged with making sure that the sessions start exactly on time. A couple of housekeeping items. Please turn off cell phones, and please remember to visit the exhibitors during the breaks because they help support all these activities. And the speakers, please keep to your time. Strictly, because we are here. So we're going to begin the program without further pause with Emerging Therapies for Motor Neuron Diseases, presented by Peter Anderson. Thank you.
Good morning. This is Monday morning, and I'm replacing Albert Ludolf, who is unavailable today. I've changed the title of the talk a little bit to Emerging Therapies for MND ALS, with a focus on the role of SOD1 as possible a new target for treating ALS. My disclosures: I have in the past 5 to 8 years been involved with a number of clinical trials and also as advisory board members for a number of pharmaceutical companies. So I have a conflict of interest for some of the things I'll be telling you today. See, the bad news in ALS we all know about is that riluzole has insufficient effect. The 1994 paper was a breakthrough paper by the French group, and it was followed up, as you know, by a large international study published in The Lancet in 1996, showing that there's a dose response in survival in riluzole-treated patients. But since then, very, very little has happened. There are more than 70 failed studies. One is, of course, the Neuron study from Japan, but that has not been proved to extend survival. Why are the results so bad? ALS is a heterogeneous condition. It's heterogeneous in etiology, in biology, in genetics. There are at least 37 different genes reported to be associated with causing ALS. These genes involve at least 5 different pathways with no common features. There's huge phenotypic heterogeneity. Misdiagnosis is common, and as some of us who have been involved in clinical trials, we are aware that some of the patients, in fact, enrolled in clinical trials had not ALS. Also, when we start treating the patients, we do it pretty late during the disease course. Many of the patients involved in trials have already lost 30, 40, 50% of the motor neurons by the time we start treating them. The outcome measures we use in trials, if it's not death, it is usually the Appel score, ALS-FRS rating scale, or some kind of pulmonary measurements. They're really brutal measurements of efficacy. And then there are no good biomarkers yet to study any possible effect on disease progression. But I would say, and that is what I will be presenting to you in the next 30 minutes, things are really changing. There are advances in genetics, immunohistopathology, and protein chemistry, which is really changing the game. Beginning with genetics. The textbook tells us that 5 to 10% of patients with a diagnosis of ALS have a familial predisposition, or at least 5 to 10% of patients report that they have a sibling, a parent, a grandfather, a cousin, etc. with ALS. And those are the patients we call familial ALS. And all the others, we lump them together and call them sporadic ALS, more or less thinking that they will then have the same etiology, or a non-genetic etiology. However, if you do molecular genetics, and by that I mean DNA analysis, and many of you who are here in the auditorium today have done that, if you start DNA screen all your patients in the clinic, the picture is very, very different. As said, we have about 35, 37 genes reportedly to be causing ALS. The first of them discovered was the SOD1 by Bob Brown back in 1993. And today's figure for number of known SOD1 mutations is a staggering 216 different mutations in this small gene of 153 amino acids. And they are found in 1 to 7% of so-called sporadic ALS patients, and from 12 to 23% of patients with a known familial predisposition, making SOD1 the second most known common cause of ALS. The most common is the hexanucleotide repeat expansion in C9orf72. That is a protein with 3 different transcripts in the central nervous system of unknown function, first found by Rosa Rademakers back in 2011, and in different populations, mostly Caucasian populations, 7 up to 40% of all ALS patients, both sporadic and familial, will be carrying such a mutation. The other genes found in ALS are very rare, and some of them are pretty common in a few populations, but not in the overall European or Caucasian populations. And it's important to remember that they vary widely between different populations. For instance, TDP43, we don't see that at all in Sweden, but it's very common in Italy, in particular, and in Sardinia. I'll talk a little about genotype, phenotype, and now with a focus on disease penetrance, because I think that is part of the key to ALS. Complete penetrance, that is, all mutation carriers will develop eventually the disease. It's a matter of time, is found in about 5-8% of cases, as said. And these are the ones easily recognized. Those are the ones where the insurance companies will pay for the DNA analysis, and those are also the families where the relatives will request genetic counseling. However, what is far more common in my experience, and since 1993 I've been following more than 400 Nordic ALS families, is what we can call incomplete penetrance. That is, there are gene carriers who above the age of 70 are unaffected. And some of them even go into their 80s or 90s, still without showing any signs or symptoms of motor neuron disease. And some of these individuals will be diagnosed as having sporadic ALS if they should develop disease like the lady down here to the left. However, there will be skipped generations for unknown reasons. And these families are far more difficult to handle. The insurance companies usually will not pay for DNA analysis in these families. And DNA analysis in these families, of course, also raises ethical issues, which David Oliver will talk about later during this session. But as I said, these families, in my experience, are far more common than the families with complete penetrance. The third mode of inheritance is, of course, recessive. It is rare in ALS, but there's one mutation, the D98 SOD1 mutation, which is common. In fact, it's the second most common known cause of ALS worldwide, found in almost all countries, including in Mongolia recently. And here you have, the good thing is that the homozygous patients, of course, their relatives will be heterozygous, and taking care of such patients, I have 11 such patients in my own clinic, is very, very easy as a clinician, because you know the cause of the disease, you know the prognosis, they always go very slowly, and the relatives do not have an increased risk of getting the disease. However, the fourth mode of inheritance, which is really a troublemaker for many of us, are these pedigrees. That is, you find not only ALS, but you also find dementia, usually FTD or Parkinsonism in different individuals, and they all carry the same mutated gene as you found in the ALS patients in these families, and you still come up with individuals like this lady who don't show any sign of neurological disease, and she's 80 or 90 years old. She plays bridge twice a week, goes to a golf course perhaps 3 times a week, but she has siblings with ALS or FTD, for instance, a child with FTD, etc. How do we explain that? We cannot, not yet. So the picture of ALS is really changing. This is more or less my desktop as it looks today. Defining the borders of ALS from a clinical point of view is becoming increasingly difficult, in particular when we need to decide when to enroll patients in clinical trials. How much will we accept of other features in our patients if we're going to enroll them in clinical trials? And that really brings up a notion, what is a diagnosis? Sir Thomas Lewis made this statement many, many years ago, and I will argue it is more, let's say, up to date now to think about this than ever before. Is a diagnosis only what we find on the EMG needle? Is it the symptoms and signs we see in the clinic, or is it, in fact, the DNA analysis or some future biomarker that decides whether or not we should consider a patient to have ALS? So the concept of what is ALS, I would argue, is really changing. When I went to medical school many years ago, it was like this on the left. During the 90s, when we found the first genes, things were changing, and we struggled to keep all the patients we found with different mutated genes inside the same envelope. And we realized when examining many patients and also doing post-mortem examinations, it's not possible. In fact, I would argue, and I've seen way more than 200 patients with SOD1 mutations. I travel around the world to see patients with different mutations in different countries, that, in fact, they are almost all of them. There will be some patients that goes outside what most of us would call ALS, and we will not always include those patients in clinical trials, though they still carry the same disease gene as patients that are well within the envelope. Turning to neuropathology, things are also changing. In Sweden, we have what we believe is the world's largest ALS pedigree, and it is really big. The pedigree will go from this wall to that wall. It's 2,200 individuals in 14 generations in 6 countries now. It's a C9orf72 family. We have collected more than 700 blood samples from members of the same family. We have 10 autopsies. And since they have the same gene, we came from the same small village in northern Sweden, you should expect the same pathology. But we don't. But what you find are the inclusions, of course, of different proteins. Ubiquitin, first found by Nigel Lee and Michael Swash many years ago, to be a hallmark of ALS, is seen in all cases, but some of them, most of them in fact, have TTP43 also inclusions. We can accept that, but some of them also have FUS inclusions. How would you expect that in a C9orf72 positive individual? But we do see that, but not in all of them. But what all of them have also is misfolded SOD1 inclusions. Both FTD and ALS patients with a C9orf72 expansion in this particular family are SOD1 positive. We don't know why, we don't know how, but that is what we find. But again, finding inclusions, is it the cause of a disease? Is it a consequence, a collateral side effect, compensatory mechanism? We don't know. But I'm going to present you some data now arguing that in fact these inclusions, including the SOD1 inclusions in particular, are the disease causing. They're not innocent bystanders. SOD1, or superoxide dismutase type 1, is a protein expressed in all cells in the human body. It is a small, very compact, robust molecule. There's 2 identical subunits, each of 153 amino acids, highly conserved. There's a copper and a zinc, and then there's an intra-subunit disulfide bond, which is critical for the stability. And this bond really pulls each subunit together. And what is very important is the SOD1 is so conserved that in fact these organisms have almost identical SOD1. That's something which these 2 gentlemen share, not only their political views, but we all have the same SOD1 molecule, unavoidable. There's one big exception, the mouse. Mouse SOD1 is rather different from the SOD1 you and I have. There are 23 different amino acids. Human SOD1 has a negative charge of -6 under physiological conditions. Mouse -11 is far more repellent to aggregate. And that, I think, is one of the critical issues why so many SOD1 trials, ALS trials, drug trials have failed. And I will explain to you why in a moment. You need to remember this for the next slides. If you autopsy SOD1 patients, this is what you see. This has been shown by many groups, that there are aggregates of misfolded SOD1 in the cytoplasma. These are missense mutations. This is a nonsense mutation, and they look rather similar. Here you have a homozygous D90A patient. And this is a very stable mutation. You have a fine granular distribution of the misfolded SOT1. But since this is homozygous, every SOT1 molecule in that individual will be mutated. These others are heterozygous. If you overexpress this mutation in a transgenic mouse model, which we did in 1996, and then follow the development of the aggregates, you can see that these, in fact, appear very early in life. And then they increase. Usually these mice get ill about age 220 days, and then we have to sacrifice them at about 250 days. So the aggregates appears very early in life in this transgenic model. However, if you use the same antibodies to stain patients without SOT1 mutations, and Thomas Brandstrom, my neuropathologist, did that back in 2002, 2003, this is what you find. You find similar inclusions containing wild-type SOT1 that is misfolded, and you do both so-called sporadic ALS and familial cases. And we see it in almost all our ALS autopsies. There appears to have misfolded SOT1 in the cytoplasma. In fact, when we do all our cases, and we have now more than 26 C9orf72 autopsies in our biobank, they all show this. Patient with FUS mutation, VAP-B, alsin, they have it. And this is wild-type SOT1 that unfolds and misfolds. And with a morphology very similar to what you see in the very stable D98 SOT1 cases. But it's very different in morphology to what you find in the unstable missense mutations A4V and D76Y as shown here, but we never find them in controls, including controls with neurological diseases. Also, of the 216 found SOT1 mutations, a handful of them has normal SOT1 activity. The mutation does not affect this mutation, that is the reduction of the superoxide free radical in these individuals, showing that it must be a gain of function that happens. So that raises really the question, is wild-type SOT1 pathogenic? Can it cause ALS? So we did the very simple experiment, or Karen Grafmo in fact did, simply overexpress human wild-type SOT1. The SOT1 you and I have in our bodies, in all cells of our body. And if you do that, those mice get ALS. That's an unavoidable conclusion. With onset of disease around 100 days, progressive loss of motor neurons, muscle wasting. And they have increased SOT1 activity, but they still lose their motor neurons. Again, showing it's a gain of a new novel function. But you could argue that we see SOT1 positive inclusion post-mortem. Could it be something happening terminally in the patient? Could be one argument. And again, do we know that they are pathogenic in vivo? So we set out to answer that question by doing the following. From the spinal cord of transgenic mice, first, we simply purified these aggregates, and we have in our group Professor Stefan Marklund, who back in 1973-74 discovered SOD type 3. That's an isoenzyme to SOT1, and we have in our university a big facility for SOT1, 2, and SOT3 research. He's an expert in purifying SOT molecules. So we did that and purified it, and then to visualize how does these aggregates of SOT1, what do they look like? We developed an assay which we call binary epitope mapping assay, which I will explain briefly to you. The SOT1 molecule is said is 153 amino acids. If you then cut it into smaller pieces, polypeptide sequences of between 15, 17, 19 acids or amino acids in length, and then you inject them into rabbits, you get polyclonal antibodies against that particular sequence. And it is so that if you have short polypeptide sequences of 12, 15, 17 amino acids in length, they don't have a defined structure. They cannot form a complex, usually. It depends on exactly which sequences you choose. But these antibodies raised in rabbits They are very specific for unfolded SOD1. They never stain. Like here, you have native folded SOD1. Because that is, as I said, a very compact, rigid structure. But the unfolded SOD1, where using EDTA, you remove the metal, and with uranium chloride, you get it to unfold. It will stain heavily on a simple Western blot with just a few seconds of exposure. So these antibodies will recognize everything which is, let's say, freely moving on the outskirts of these aggregates, but what is buried in the central part will not be seen. So in the binary epitope mapping assay, we can detect what is on the outside of these aggregates. And we did this then on aggregates purified from 3 different human mutations found in ALS, the D98, G93A, and G85R, and compared also with wild-type human SOD1 overexpressed in mice. And this is what we find. We find that 3 of these, the G93A, G85R, and wild-type human SOD1, they have a very clear pattern of antibody response to these aggregates. And that is what we call strain A. However, the D90A is radically different. You will see there's a big antibody response to these epitopes. This will be these antibodies to these areas here. And this is very interesting because this is part of the periphery of a molecule, usually, but it is in a different shape compared to the wild-type SOD1. Here you see there's no response to wild-type SOD1, but the D90A has a very clear antibody response to this area, showing that in D90A aggregates, these amino acids sequences here are exposed on the surface of those aggregates, done by dot blot assays. So, and this is what we call strain B. Then we tried in the lab, is it possible if you purify SOD1 from red blood cells, for instance, just wild-type SOD1, can you mimic these aggregates in the lab by changing the pH, for instance, by increasing the temperature, by oxidizing or reducing the disulfide bond, etc.? Can you, let's call it, mistreat SOD1 and get aggregates with the same conformation? And this is our results, eight different conditions, and it's not possible simply by simple chemistry to get human SOD1 to misfold in vitro as it does in these transgenic mice. This shows that the aggregates that are formed in the transgenic mice, their shape is very much determined by biological processes in the central nervous system, which we don't understand yet. So it's not a coincidental finding, these different shapes. This is very robust finding, and there must be a biological cause for that. Then we looked more closely at the G93A SOD1. The G93A mouse, developed 1993-94 by Mark Gurney at Harvard, is the standard transgenic mouse model for studying ALS. That is the mouse model available freely from Jackson Lab, which has been used in so many clinical drug trials to screen for possible drugs that will work or not work in ALS. And this is why this is the worst possible model for ALS. There's no worse model than the G93A. And this is what you see on this picture. Because if you study these mice, and they have usually 20 to 30 copies of a human G93A SOD1 mutation, what you find a huge amount, not large amount, a huge amount of misfolded SOD1 aggregates in these mice. Usually these mice will develop symptoms around the age of 85, 90 days, and they will live for 3-4 weeks before you have to sacrifice them. And most of the clinical drug trials done in these have done around symptomatic onset here, and in some papers they will, for instance, the arimoclomol paper, they did it pre-symptomatically, treating at day 35 or 50 days of the life of a mouse. But already at that time, their spinal cord is packed with misfolded SOD1. please notice that this is logarithmic. So there's an exponential increase in SOD1 aggregates in the spinal cord of G93A mice, showing that this is a catalytic process. Something is happening, causing a huge formation of these aggregates in these mice. And therefore, this model should never be used. It is not a reflection of human SOD1, and there are far better SOD1 models. I will show them to you briefly. So this suggests that what happens in the G93A and also in other models is that you have a SOD1, and when you goes into the unfolded stage, which then misfold, forming oligomers, fibrils, and these large fibrils are the ones we observe post-mortem. And this process is catalytic, because you can see the way it increases. However, if we then want to test, are these pathogenic? Are they really causing the disease? The logical thing is, let's inject some of this into the spinal cord and see what happens. This is exactly what we did. We simply add some more of this, and can we then move the process forward? And the answer is yes, we can elicit ALS now by injecting misfolded SOD1. So what we did is simply, this is the setup. We make a small micro-injection here at the L3 lumbar spinal cord, about 1 microliter corresponding to 5 nanograms of SOD1. That is a very, very small amount. And we do it in the left ventral horn. This is showing that we hit the right target. And now comes the second problem. Not only hitting the place, but which animal to use. And as I said before, mouse SOD1 is very different from human SOD1. So mouse SOD1 cannot be used as an aggregate or a substrate for the formation of SOD1 aggregates. It won't work. You have to have overexpression of human SOD1. Wild-type human SOD1 won't work for kinetic reasons, because it is pretty stable, and in a short lifespan of a mouse, it will not be feasible to use. You have to use a human SOD1 which has a higher propensity or likelihood of unfolding and misfolding. And we choose the G85R SOD1 transgenic model developed by Don Cleveland and Lucy Bruin, because it has, and this is shown here, it has a very long lifespan when suddenly the disease begins and these mice forms SOD1 aggregates in the spinal cord, but there is a span of more than 300 days where there are no SOD1 aggregates in the spinal cords of those mice. And this is where we want to inject our SOD1 aggregates purified from spinal cords of other mice. And this is a busy slide. On the left, you will see we do the injection at 100 days of age, where we inject strain A SOD1 aggregates purified from other mice, and 60 to 70 days later, they will have onset of a disease in the left leg because we do the injection in the L3 ventral horn, and they will survive for a few weeks, and then they will die. If we then inject strain B, purified from the spinal cord of D90A overexpressing mice, we get a little bit later onset of disease, but it's the same pattern. If you then look at the time course at different times after the injection in the mice, you will see that there is increasing accumulation of SOD1 aggregates with the same conformation as the one from the donor mouse in the recipient. So if you inject strain A, you get strain A aggregates in the recipient mouse all through the neuroaxis. More importantly, if you inject strain B, that is from the D90A mouse, into G85R mice, which usually do not develop strain B aggregates, you can force the G85R SOD1 to form strain B aggregates. Did you get that? So you can push a G85R mouse, which should form strain A aggregates, to instead form strain B aggregates. That means, again, it is a highly controlled catalytic templated process that takes place. And this is really the proof of the concept that the formation, the conformation of these aggregates are disease-causing, at least in mice. So strain A injection in the seed caused strain A aggregate formation in the mouse. Strain B injection caused strain B aggregate formation in the recipient mouse. And again, as said, the G85 usually do not form strain B aggregates, but if you inject strain B aggregates from a D90A, it will, showing that it's not the mutation that decides the conformation of the aggregate, it is what is pushed into it. We do a lumbar injection, and here you can see how it progresses rosary through the central nervous system of the mice. There are differences between strain A and B. And again, strain A reaches more into the brainstem and even up to the brain and a little bit to the cerebellum, but note there is a different axis here. However, strain B goes a little bit slower, and it has far, far less in the brainstem than strain A. There are phenotypic differences decided by the conformation of the aggregates. And this is very interesting from a clinical point of view, because D90A homozygous ALS patients have always lag onset, and they have only late in the disease bulbar symptoms. So this is very much a lower spinal cord disease. So coming to serial injections, if you do multiple injections in the same interval, you can preserve the conformation. There's a dose response. If you increase the dose of the aggregates, you get earlier disease. Now comes to human aggregates. And these are the last slides I will show you. G127X purified from a human autopsy causes disease also. This is a G127X overexpressing mouse. Of course, here you have lots more SOD1 there, so you get earlier disease. D90A is the same, but here we had 3 patients where we had autopsy material available, and one of them did not show, but that patient survived for 27 years with ALS. And again, there were very few cells left in the spinal cord where you could purify the aggregates from. Now, a patient with a FUS mutation. This is very provocative. FUS mutation, FUS is a RNA housekeeping enzyme. I'll be done in just a minute. And this was a very tragic case I had in my clinic, a 16-year-old girl, teenager with ALS, progressive bulbar onset type of ALS, and with this FUS mutation. And she died after 8 months. She had no other diseases whatsoever. And her spinal cord motor neurons were full of misfolded SOD1. We looked for SOD1 somatic mutations. There was none. The SOD1 activity was normal, but there were lots of misfolded SOD1 aggregates all through her motor system. And we purified that, injected it into 6 different mice, and this is what you see. They elicit ALS disease in the recipient mice, propagating all the way through the spinal cord of those mice, again causing misfolding of SOD1 with the same conformation as we observed in the patient. Again, strongly suggesting that misfolded SOD1 aggregate plays a role in FUS-induced ALS. We're also doing injections in other parts. I want to get to the last slides. So what happens, we believe, is the following. You have the scenario up here to the left. That is normal. The SOD1 dimer can dissociate into monomers. That's a normal process. Sometimes it will unfold. That is still reversible. But this can be pushed into formation of different form of misfolded SOD1 aggregates. 1 or 2 of these, perhaps 3, can form these prions. And when injected, they will cause disease. But these constitute only a very small amount of a total amount of SOD1 in these animals. And this is the targets for new therapies for ALS. So what we need to do in the next 5 to 10 years, and I'm particularly saying this to the industry, if they are present, we need to reduce the amount of SOD1 that is being produced. We need to stabilize the SOD1 so it cannot unfold and misfold, or we need to block the SOD1 aggregation so it will not cause disease and the formation of these prions. So there's, in fact, light on the horizon. This is my university campus, what it looks like. In the future, we'll have to do CRISPR-Cas9. But there are also bad news. I need to say that very clearly, because you cannot bring down the SOD1 too much. SOD1 is an essential enzyme. You need to have at least, in our experience, 15 to 20% of SOD1 activity to protect against free radicals, which means that you have to initiate treatment both very early, and you need to combine treatments because you need to have some wild-type SOD1 available to remove free radicals. With that, I thank you for your attention.
Thank you. Thank you very much for an excellent presentation. The presentation is open for questions. Please come up to the microphones to ask your questions. To the right. Do you have a question?
Yes. We know that there are some controversies between the use of statins and the development of ALS, but I'm not sure. There's a paper showing that in the mice model of SOD mutation, the mice, statins can have a protective effect in the development of symptoms in the mice model. I'm not sure if it's...
So you're asking about statins as a risk factor for ALS?
Yes, in the mice model, SOD mutation. It shows a protective.
But then the first thing I would do, both as a reviewer, if I was reviewing such a paper, I would look which mice model did you use, because I would, based on all our results, and we also looked at other models that I presented here, we've had more than 30,000 transgenic ALS mice in my laboratory in Umeå. Most of those studies, they do not stand up to quality control in my experience. And we have not tested whether statins affect the formation of SOD1 aggregates. We haven't had the time. Perhaps Leonard van den Berg can comment on that in his next presentation. I think you know more about statins and ALS than I do. I think this...
Yeah, you mentioned families that carry pathogenic mutations in SOD1 that show incomplete penetrance. Is there any DNA sequencing efforts to try to identify the genes that explain the resilience?
Could you please repeat?
Genes that, are there efforts to identify the genes that could explain the resilience in the families that show incomplete penetrance?
Okay. The question asked by this gentleman is the cause of reduced disease penetrance in SOD1 families. A very good question, and in fact, focus on my own personal, let's say, hobby research. There are some SOD1 mutations that consistently go with reduced disease penetrance. The prototype is the I113T, which is the fourth most common. It's the most common one in the UK. And the mutation next to that, the G114A, is always complete penetrant. So you have two different codons, one with always reduced complete penetrance and one with complete. And we have no explanation for that either at biological level. We and other groups have done both whole genome sequencing to find are there additional genes modifying that. It appears not to be the case. And since there are many I113Ts, almost always showing the same pattern, I think it's something with the particular mutation and its effect on the misfolding. And that's why I brought that up initially, that these mutations that are associated with reduced disease penetrance, that could be the propensity for the molecule to unfold, misfold, forming of prionic species. So it will unfold, but will it also misfold, irreversible, forming prion strains? And I guess that that is a stochastic event. There's a certain probability that prions will be formed. There will be formed aggregates, but are they prionic aggregates? Not always. Sometimes, but not always. That would be my speculation, but I don't have proof of that. I never got an I113T autopsy. It's very difficult to get ALS autopsies today, as some of you may know.
I'm just wondering, can you make a second site mutation in the SOD1 that prevents the actual formation of aggregates?
That's our dream. We're doing a study, EU funded by the way, where we're doing mass screening of old healthy people in 6 different European countries, and we are looking for SOD1 mutations in old, healthy people, mutations we never find in ALS. And so far, we haven't found it. But theoretically, it's a fantastic question you ask. That is, theoretically, there should be a possibility of SOD1 mutation that should prevent ALS. That means a mutation that will flip the molecule, let's say, to the other side, so it cannot form prionic species. As some of you may be aware, that has recently been shown by the group at Queen Square that, in fact, the prion protein, there's a mutation where I think it's a British family even, where there is a mutation which will prevent the prion protein from forming prions for Creutzfeldt-Jakob's disease. And theoretically, we think that's the same in SOD1 ALS. We just haven't found the mutation, but I guess it will be in codon 32. Tryptophan 32 will be the one I'm looking for. But we've so far screened about 1,200 old, healthy people, and we haven't found anyone. It's a big, big task, but we're continuing working on it.
One last question. Here.
No, right here, okay. Thank you very much, Peter. And I like just to ask you if this SOD model and from your work is the same concept could be imagined or applied for the other mutation like FUS or like TDP43, and is this seeding system will be exactly the same or not, or that's only the SOD process is only for SOD mutation or a general process in ALS?
See, and this is really sticky because if there are more than 37 ALS-causing genes, SOD1 is one of them, C9orf is the second one. We don't have autopsies from all of them. We don't know if SOD1 positive aggregates are formed in the spinal cords of all of them. The ones we have had access to, we see it, in particular in the C9orf72. So, in other words, if you develop a new treatment targeting SOD1, that treatment may also be of value for patients with C9orf72 mutation. But you cannot exclude the possibility that during the disease process in a C9orf72 patient, that there will be additional effects on other proteins that may also be damaging to the central nervous system. SOD1 may be only one pathway. I can reveal to you, we don't have that published yet. We have a manuscript submitted. In fact, at least in one type of ALS, there appears to be 2 different strains working in parallel. So if you only target one of them, the other one will still kill your patient. So you need to hit both of them. But that is very preliminary data I did not show here for lack of time.
Okay.
It's a very complex story.
Thank you. So I will now call Leonard Vandenberg for clinical concepts in ALS. So Leonard Vandenberg is professor of neurology in Utrecht, Netherlands, and is director of the ALS, the Netherlands ALS Center, and he is also the chairman of the ENCALS, so that's a European network to cure ALS. That's a vast program. And thank you very much to be there, and go ahead.
Thank you, Claude. No mice in my talk. I'm vegetarian. Not out of free will, but my wife and daughters became vegetarian, and my life is more peaceful not to resist them. Just to start, this is a cow in the Netherlands. The Netherlands is 60% below sea level, and that's a cow in the mountains here in Austria. So that looks much nicer here. But the main message is, my last name is Vandenberg. It's the most common, one of the most common names in the Netherlands, and it means from the mountain. So I'm very happy to be here. Thank you for the invitation. The title, the clinical concept of ALS, I think it's a very interesting title. I think we cannot underestimate that, that we learn a lot from the patients that we see in our clinics, and to phenotype them and to apply all the technology that we have now to samples that we obtain from patients, I think will become more and more important in the coming years. Applying exciting technology in genetics, in imaging, but also in biomarkers and in the stem cell technology, where we're able now to generate models from fibroblasts from patients, so human models in a dish, so human motor neurons in a dish. So very much patient-derived. The research that we do is very much patient-driven. These are 3 patients from our clinic, Bernard Muller and Robert John Stout, who started a company, Treeway, to discover drugs for ALS, and they came up with an oral form of edaravone, and they're trying to develop that now. It's Harm van Soest, who started an ALS investment fund to spread the risk for investors in ALS, and that fund contains now over 20 million euro, and is able to fund trials for ALS. And they started Project Mine, a large genetic project I will talk about later. What is ALS? ALS is a disease of upper and lower motor neurons. Onset is usually mild in the beginning. 1/3 of the patients start with dysarthria, 1/3 with weakness in hands, 1/3 of the patients with weakness in legs, and the minority of the patients start with respiratory problems. The median survival, it's always a progressive disease. The median survival is 3 years after onset. Onset of disease can occur at any adult age. The incidence of the disease is 2 to 3 per 100,000, about the same worldwide. Over 250,000 people die of ALS yearly. 10% of the disease is familial. 5% also have frontotemporal dementia, and 30 to 50% of the patients have impaired cognition. There are no remissions, no cures, and no effective treatment. So it looks all very simple and very homogeneous, but if we really look at all these features, they're very heterogeneous. And I think in ALS research, the heterogeneity, solving the heterogeneity of the disease is the challenge for the coming years, to find and discover better treatment for this devastating disease. If we look at loss of upper and lower motor neurons, we all know in clinic that some patients have only lower motor neuron symptoms, and they're diagnosed with progressive muscular atrophy, and other patients only have upper motor neuron loss and are diagnosed with primary lateral sclerosis. It's very difficult to measure the extent of upper motor neuron loss. Well, for the lower motor neuron loss, we have the EMGs. But exactly where is the cutoff? When do we decide whether a patient has PMA or PLS is difficult to determine. I think this is an important paper from 2006 in neurology by Gordon, the group of Columbia, and they showed that if you do a follow-up in patients who present just with upper motor neuron symptoms, that within 4 years, most of the patients develop ALS. Actually, 77% of the patients developed lower motor neuron symptoms by 4 years from symptom onset. And they concluded that the diagnosis of primary lateral sclerosis cannot be made prior to 4 years of onset of symptoms. And we have done similar research and came up with the same conclusion. And we've also done quite some research with Amsterdam group on lower motor neuron disease. And I think the same is true for patients who present initially with lower motor neuron symptoms. We did some follow-up studies up to 5 years in patients, in one group of patients that had a disease duration of less than 4 years, 37 patients, and 49 patients who had a disease duration of longer than 4 years. And most of the patients with disease duration of less than 4 years developed upper motor neuron loss within 4 years. and the median survival was 56 months, which is a little bit longer than ALS, but still most of these patients developed an ALS phenotype. While the patients who had a disease duration of more than 4 years had, if they had, usually when they had a segmental distribution of weakness, so weakness in arms, in the hands, or in more proximal muscle groups in the arms, when it was still segmental after 4 years, those patients had a very benign disease course. So the message is that it's not possible to diagnose patients with progressive muscular atrophy or primary lateral sclerosis within 4 years after onset of symptoms. So that's sometimes difficult to tell the patients that we're not sure of the diagnosis yet and that we have to do careful follow-up in our patients. If we look at the onset of mild weakness and the progression of the disease, it's also very heterogeneous. This is just a graph from the lithium trial, from the LICOS trial in the UK. You can see that the variability, but also the distribution of the functional rating scale is very, very heterogeneous over time and between patients. We looked in our patients where there is a pattern of symptom development in patients with lower motor neuron disease. And we sent a questionnaire about the symptom development in 470 patients. 254 patients had ALS, but we also asked patients who just had lower motor neuron loss and patients who had just had upper motor neuron loss, just to see whether there's a difference in the pattern of symptom development in the lower motor neuron or the upper motor neuron. And what we found is that the onset of the disease is very focal in most of these patients, 85% of the patients, and that it's very, very likely, that was highly significant, that if you develop weakness in one hand, it's very likely that the next weakness will occur in the other hand. The same is true for the legs. If it occurs in the left side, it will, the next limb that will be affected will be the right leg. And that was true in ALS, but also in the lower motor neuron phenotypes and the upper motor neuron phenotypes. So in patients with limb onset, symptoms occurred significantly faster in the contralateral limb, followed by the other limbs, and lastly by the bulbar regions. So the preferred spread of the symptom from one limb to the other and to adjacent sites suggests that there is a symptom spread is organized and possibly involving axonal connectivity, and that's also now a hot topic in pathology. That it's, and Peter already talked about it, about the prion-like distribution of the disease, and if you translate that to how we think the disease spreads, it could be a prion-like spread, but it will start focally, and then first it will go through the corpus callosum to the other side, and the same is true for the lower motor neuron, and then it will spread to adjacent areas, regions. But there's always an exception, and we also found that there are also patterns of non-contiguous symptoms development. So in patients who had onset, a bulbar onset, 30% the weakness was followed by symptoms in the leg, and the same is true for patients with leg onset, that the next weakness would be bulbar symptoms in 11% of the patients. And we looked at whether there were some differences between patients with a contiguous symptoms development and non-contiguous development, but there's no difference in age or other characteristics of the disease. The median survival is 3 years after onset, but we all know that that can be very different, heterogeneous too. Some patients die within a year. We all know Stephen Hawking lived with ALS over 50 years. So, and 20% of the patients live longer than 5 years. So we thought whether it would be good to have whether we could predict that better. Now we tell the patients you have ALS, the median survival is 3 years, and it's difficult to tell how the course of the disease will be. So we tried to develop a prediction model, a personalized prediction model, so that we could predict survival in individual patients, not in groups. We published that in the Lancet Neurology a month ago. And why we thought that was good is the advantages that you can have personalized care. It's important information, the prognosis of the patient for the care pathway, also for timing of interventions. But you can also use this data for inclusion and stratification in trials, or to identify particular subgroups with better or worse prognosis, for example, for trials. The methods is all in the paper, so if you want more detailed information about that, you can read it in the paper. Data collection, of course, is the first step. We spent some time on the missing data and the variable selection. We used the most innovative methods for internal and external validation, sensitivity analysis. And at the end, when we had the model, we also thought, together with ethicists, how to implement the model. Do we have open access of the prediction model to patients or only to healthcare professionals? These are we started with 16 variables: gender, site of onset, age at onset, definite ALS, diagnostic delay, vital capacity, the ALS-FRS slope, premorbid BMI, current smoking and cigarette pack. Yes, these are all predictors from the literature that are known in literature to play a role in survival. We looked at some characteristics of cognitive function, and we also looked at the effect of the C9orf72 repeat expansion and the UNC13A SNP, which also plays, we know that the CC haplotype has a worse prognosis. If you do multivariate analysis, we ended up with 8 variables: site of onset, age at onset, definite ALS, vital capacity, the ALS-FRS slope, the presence of FTD, clinical diagnosis of FTD, and the C9orf repeat expansion. We used registries, patient registries from 9 countries, 14 data sets, and all together we had data from 11,475 patients, a very large data sets on detailed information about the phenotype of the patients. And these are the largest data sets, and these are also population-based studies, the population-based study in Italy, Ireland, and the Netherlands. I won't go into detail much about the methodology, but the principle is that you do external validation, very important. You take one country, you compare it with the other, and that you do with all possible combinations. So then you take the other country, compare it with the other 9 countries, and you do that over and over again. And then you add up with a prediction model, which was proved to be valid and generalizable to the whole set of patients in different countries. This is an example of 25 patients, just to show how reliable or not reliable but how precise the prediction can be. So this is 25 patients. Here are the 8 variables. They are different for each patient. And this is the black dot is the median. The colored part is the interquartile range, and the bar is the whole range. And what you can see is that patients who have a short, we're quite precise in predicting the prognosis in patients that have a relatively short prognosis, short survival. But patients who have a longer survival, the prediction becomes a little less accurate. So it's between 5 to 10 years, of 7 to 15 years. But still, it's a lot better than just say that the median survival is 3 years. And what also can be helpful if you don't want to tell the patient the precise prognosis for each individual, we also grouped patients in 5 groups. So patients with a very short prognosis, short, intermediate, long, or very long. So you can also group it in 5 patients. We asked patients whether they want to know the prognosis. Would you, if we can predict your prognosis, do you want to know? And so these are all Dutch patients, so that it can be different in different countries. But over 60% of the patients, I was a little bit surprised, but I think still a lot, over 60% of the patients do want to know a more precise prediction of their survival. 20% do not want to know, and 20% did not know whether they wanted to know. We talked to ethicists and decided that we would make the model available only to healthcare professionals and not to patients, because we thought that patients may regret maybe that they know the prognosis after they've done this, and they probably need more time and more explanation of the model. And so maybe we will do that later, but it's now not open for patients. So you can just sign up if you want to use it in a clinic. That was the previous slide. This is a clip, which usually, it's almost a rule, usually don't work. Yeah, there it goes. So it's kind of, you can put in all kinds of numbers. So that's the ALS-FRS. If you reduce that, then the prognosis, predicted prognosis, will go down, and you can add all kinds of, you can add all the information of the 8 predictors that we have, and then you get a curve and additional information for the patient, for an individual patient about their prognosis. I think it is useful for inpatient care, for care pathways, to decide when you will talk about devices for patients or other like invasive ventilation or decisions around end of life. You can use it for, we can probably use it for, to identify subgroups for trials, certification, and inclusion rules in trials. I think these prediction models for all kinds of, also maybe for function, will become more important, and it's very important to collaborate well between countries and registries, and we're doing well in Europe about that. So we came up with a reliable, generalizable, and easy-to-use prediction model. So please go to the London Neurology and if you want to use it, or just go to the site www.encalspredictionmodel.com. When we were submitting the paper, Stephen Hawken died this year, as you all know, probably one of the best known ALS patients. And there was lots of debate whether he actually had ALS because he lived so long. So we decided to apply what we know from his disease to our prediction model, just to see whether it was unusually long. So we tried to see whether his prediction was as rare as his intellectual performance. When he was actually diagnosed in 1963, and he was told that his survival was going to be 2 years. So that's quite different from his actual survival. So we applied, or we found in the internet, the 8 determinants were kind of easy to find, also from the movie on his life, the 8 variables, and we applied that to the model, and his predicted survival, median survival was 30 years, so all on the extreme side of the cursor. These are actually all the 11,000 patients in our combined registry. So he had, based on his 8 predictors, he had a median survival of 30 years. And if you go to his prediction probability curve, his median survival is here. He got his tracheostomy. So the endpoint of the prediction is a combined endpoint. So it is death or tracheostomy or being on a non-invasive ventilation for more than 23 hours a day. So actually here was his endpoint when he got his tracheostomy. And so if you look at this predicted survival, his endpoint was quite early in the disease. He could have lived without tracheostomy, according to the prediction model, quite much longer. And he lived about 30 years longer than predicted because of invasive ventilations and intensive supportive care, but that will be the subject of the next speaker. And, but still, when he died this year, he still had a 20% probability, survival probability. And when he diagnosed, for example, he had a 94% survival probability of 10 years. So he could have been much better informed. We sent this letter to the London Neurology again and it was accepted. We thought we had this very nice title, Acceptable Scientist with a Predictable Life Expectancy. Took a long time. We thought it was a good title, but they changed it to the life expectancy of Stephen Hawking according to the NCALS model. It's quite a boring title. It's not. If there are any, we like. If there are people here from London Neurology, we like your journal. Don't worry, but the title could have been much nicer. Actually, I read something about an interview of Stephen Hawking last night, and he said that he did so well in his research, and he worked so hard because his survival was predicted to be just 2 years, so he worked harder and harder. So maybe it's good that this prediction model was not available in 1963. I come up with these ideas, maybe I should tell my PhD students that, well, I won't do it. Okay, then the onset can occur at any adult age. So my youngest patient is 18 years old, my oldest patient is 94 years old, and I remember when I diagnosed him, he was 94, and I said, well, you have ALS, I'm sorry to tell you, and he said, oh, doctor, so I'm not going to be old. So that's Dutch humor. And the incidence is 2 to 3 per 100,000, and 1 out of 300 people die of ALS, so maybe 1 out of the audience here will die of ALS. So that's still, it's not a very rare disease, and if you look at the incidence, and this is from our population-based study in the Netherlands, at any adult age, the peak is at about 70 years, more in men than women, and what's interesting is that that's different from Alzheimer's disease and Parkinson's disease, where the curve goes up with higher age. There seems to be some protective factor in people with older age. So it's not just a disease of aging, but and I don't think we miss these patients because it's such a devastating disease they will not end up in a nursing home without diagnosis. So there's something here that's interesting. The same pattern is there for the prevalence. The lifetime risk is 1 out of 300. 5% have FTD, and 30 to 50% have impaired cognition. So it could be two extremes, ALS and FTD, in a spectrum of maybe a single disease. 5%, 10% have ALS and FTD. 30 to 50% have some form of impaired cognition or behavior, executive functions or behavior in ALS. And also patients with FTD, about 40% of the patients have some motor neuron, some evidence of motor neuron loss. So I think, and we started that only 5 years ago, that testing for cognitive and behavioral impairment is part of the diagnostic workup. We have now a psychologist at our outpatient clinic when we do the diagnosis on one day. We started that 5 years ago. I'm a little bit embarrassed to say that we did not start that much earlier. It's very surprising what you see. You think a patient is normal. If you talk to them, you think, oh, it's just no impairment. And then you do the test, and there's some conversation with the spouse and the relatives, and you really hear what's happening at home in terms of behavior, but also of executive functions. So it is important to do proper testing because it's important to inform the patients and the relatives. But it's also important for the health care professionals and the care pathway you initiate. It's also important because the C9orf mutations and TBK1 mutations are more frequently in patients that are cognitively impaired. They have a more aggressive disease. There's non-compliance with treatment recommendations. And, I don't have to tell you, it is a very much increased caregiver burden, and it is already quite a burden, ALS. If it's ALS and FTD, it's very, very, very intensive. 10% is familial. The term familial ALS is quite vague, and part of it is because you have to very carefully go through the family, not only for ALS or progressive muscular atrophy or primary lateral sclerosis, which could all be part of the familial ALS spectrum, but also for FTD and probably also for other neuropsychiatric diseases. There's a study from Ireland showing that in first and second degree relatives, there's also an increased prevalence of psychosis and autism, and there's also some genetic association from the GWAS studies from schizophrenia and ALS. The number of genes are increasing, as you can see here, also because of this project that we started, whole genome sequencing project in ALS, Project Mine. The major genes, as you heard in the previous talk, is C9orf, SOD1, FAS, and TDP43. We don't have any SOD1 mutations in the Netherlands, also not in Switzerland, and not in Ireland. But 1 out of 20 patients with sporadic disease have C9orf, and 30–40% of our families have a C9orf mutation. So we may need to reclassify ALS. That's also a message of what I want to say now. We do this usually based on symptoms. So do they have lower motor or upper motor neuron loss, progressive disease? Do they have onset in the bulbar region or in arms or legs? We may have to abandon this classification and go more towards a data-driven classification. We know that in families with ALS, so the same cause of the disease, so the same gene mutation, that all these phenotypes are present. So we may have to reclassify our ALS in the future to identify better treatment. Now we're doing it on symptoms, but we may have to do it based on genetics, environmental, lifestyle risk factors, imaging, biomarkers, and maybe some translational science. And we may have to identify better clusters that are more homogeneous, that may need specific treatment and useful for clinical trials. Genetics, what's the component of genetics in sporadic ALS? Well, we calculate from twin studies that the heritability, so the contribution of genes is 60%, about 60%, but the environmental lifestyle component is 40%. So it's very important to go for the genes for all kinds of reasons I already discussed, and that's why we started Project Mine, a very large genetic project, international project, to identify all common and rare risk factors, to establish a database for the years to come on genetics, a human genome project for ALS, and the aim is to whole genome sequence 15,000 ALS patients and 7,500 controls. And it was a very good project for funding, swim for a genome, run for a genome, all kinds of actions. We have now many partners, international partnering in Project Mine. Go to the website Project Mine. It's a collaboration between all kinds of research groups in all these countries. But the main message is, so far we have been able to generate funding for genotype genomes of 12,000 patients with ALS and also 8,000 controls. And it's important that this data is publicly available to a large extent and can be shared for other genetic projects. You can go to the data browser, Project Mine data browser, and you can just fill in your favorite gene. You will get all kinds of information, association with ALS, but also expression data for that specific gene. We also look at, this is my last slides. I'm a liar, many more slides. We'll just say, no, 2 minutes. 2 minutes from now. Yeah, Peter was late. He was very late. I apologize for Peter. Yeah, it's all these mice. We also look at lifestyle factors, and this is important. So Netherlands is a good country. We're highly densely populated, but we're completely flat. So it's easy to drive around, to go to our patients and draw blood and fill out all kinds of questionnaires about lifestyle, medical history, and also diet. And we were able to harmonize these questionnaires in Italian and in English, and the other population-based studies. Just a summary of the risk factors that we have now is smoking is a risk factor. So intake of alcohol is actually protective for ALS. Head trauma is a risk factor. Recent findings are that exogenous estrogens and progestogens, so oral contraceptives, are protective in women. Occupational exposure to silica is a risk factor, and also long-term exposure to traffic-related air pollution. Another topic that comes up, and that's also an answer to the statin question, is that higher physical activity, leisure time and occupational activities is a risk factor. So patients seem to be more healthier ALS patients otherwise. But on the other hand, the pre-symptomatic total daily energy intake is higher, and there's also more fat, total fat, saturated fat, and cholesterol in the diet of patients before they develop ALS. So, but the BMI is significantly lower in patients, so that's an indication that there's something going on, some hypermetabolism going on, and that's an interesting topic to study further. This is also important to say, all these risk factors doesn't mean that there is the phenotype of the patient. It's just this running patient who eats a lot of hamburgers, who goes on his bike close to cars, and then you have this protective phenotype who drink, eat vegetables, and have a higher BMI. But all these risk factors are independent, so they play a role in different patients. So we have to reclassify, we have to go to precision medicine to integrate all the data that come out from genetics, environmental, lifestyle factors, imaging, and that will be the future for ALS, to find a better treatment. And the only one cluster, that's the last slide, is the C9orf mutation. It's very specific, so we already have one cluster, we have to find more clusters. One cluster is the C9orf mutation, it's associated with FTD, it's a more rapid disease course, so shorter survival. The carriers, the pre-symptomatic carriers, have abnormalities in the brain, and also there's a different pattern of, it's a more progressive disease in the C9orf patients compared to the non-C9orf patients. They have more gray matter and also more white matter abnormalities, and also during follow-up they have a more aggressive disease. And they also have, if you look at the BMI before onset of the disease, if you compare the C9orf patients with the C9orf negative patients, they have a lower BMI throughout life. I think that's also interesting. And this is the very last slide. So it's a phenotype, I think, in ALS researchers. We talk too long, maybe. So that we're going to treat, hopefully, ALS. We're going to not hopefully, but we're probably going to treat not ALS all at once, but usually in clusters, in subtypes. And I hope that the first studies are coming now with treating C9orf with antisense oligonucleotides or maybe gene therapy. So I think that the C9orf related to ALS is going to be the first clusters, first type, first patients with ALS that hopefully are going to be treated like the exciting results in SMA. Thank you.
Thank you, Leonard. We have time for 2 questions.
Can I ask one? So can I ask a question? Does this mean that in your country you can't use the SOD model mice work from Sweden, and that every country with the different predominant genetic mutation is going to have a different specific therapy?
Yeah,
rather than the same mechanism in 2 countries? Is this what we're talking about?
Yeah, well, it's gene specific, the treatment. So we don't have any SOD1 mutations. So we are not involved in the antisense therapy studies in SOD1, but we hopefully will be with C9orf.
Thank you.
C9orf is also in the Asian countries. It's not present almost. So it's very country specific. And in Belgium, for example, 20% of the families are SOD1 positive, and none in the Netherlands.
Right.
Well, we are so close.
Interesting. Any questions?
No questions.
Yes, you described the disease as ALS with different forms, different concepts, and so on. But for you, it's one disease, or it's multiple different diseases?
Yeah. It could be like polyneuropathy. One phenotype, 300 different
Like
causes.
CMT?
Hopefully not, but I think we have considered ALS as one disease for too long, and we have to abandon that.
Because my feeling is that when we use the term of ALS for this patient, it's like if we were using the term of myopathies for all the myopathies, you know, and with no distinction between dystrophinopathies and sarcoglycanopathy.
Yep.
So maybe we have to change the classification.
Yeah.
Thank you very much.
Thank you. So our third presentation will be Professor Oliver, who's a professor of palliative care, and he'll talk to us about ethical issues in ALS.
Thank you very much, and thank you to the organisers for asking me to speak today. I'm moving from heterogeneity of diseases to heterogeneity of people and families. What are the sort of ethical issues that we face? We have the problems, I think, of communication, particularly as we've heard about genetics. There are things about bulbar issues, of cognitive change, of ventilatory support, of end-of-life issues, and increasingly discussions about hastened death. And I'd like to look at all of those as we go through. If the slides will move. I'm pressing the button and nothing's happening at the back. Could you? Oh. When we're talking about telling the diagnosis, how is that done? We know from research, particularly in Australia, that many patients find being told the diagnosis very unsatisfactory. 35% of patients said that it was very poor, and that also that neurologists find it very difficult. And in the NICE document, which is the National Institute of Health and Care Excellence, we did a review of MND-ALS care. And within that, we made recommendations that there was a need to be truthful, which I hope we all would be. But the diagnosis should be given by someone who knows about MND-ALS. And at that time, there may be the opportunity, the wish of patients and families to talk about the prognosis, to talk about treatment options, what is likely to happen, and what are the likely symptoms. And I think it's important that that comes from professionals. I know of one patient who was told in a very good way about their diagnosis, came out into the reception area where someone said, here's the details of the MND Association. You'll need them when you get your wheelchair. And don't look on the internet because it always talks about choking to death. And that patient, till the day they died, was thinking of choking to death. And it's needed to have good information. We need to look at patient wishes, but there are also the concerns of families, and we all face families not wanting to tell the diagnosis because there are fears of the patient, there are fears of the family, and there are fears of the professionals. And I think now we do have what do we say about genetics? As has been said, many patients will have a familial form, and increasingly we know that other patients who have sporadic may have abnormal genes. And I think there's real ethical debates of how we discuss that. It's really complex. For the patient, I now know that my family are at risk. For the families, I'm now hearing I'm at risk, but my family is at risk. How will it be? Because patients start thinking, am I going to be like my father, my mother, my uncle, my cousin? How do they tell others within the family? Do I tell my brothers and my cousins? How do I tell my children? And I think for the professionals, we're faced with very big ethical issues about confidentiality. When families ask what's happening, but the patient is saying, don't tell them. And we are facing patients who may have cognitive change at the same time. So have they got the capacity to deal with that information? Real big ethical issues. And then when we talk about bulbar problems, we've got problems with swallowing, with food and water and medication. And associated with that is drooling and obviously communication. So how do we discuss and talk about the ethical issues of swallowing? When do we talk about gastrostomy? How do we talk with patients about a gastrostomy? Because we know there are benefits. There are probably benefits of quality of life. There may be, although it's debatable for survival, it's certainly anecdotally, I find that it allows people to remain at home because we can give medication and perhaps hydration at the end of life. But there are risks. You know, there are risks of the procedure. There's risks afterwards. There's risks of how a gastrostomy is coped with as someone deteriorates. And we may need to talk about gastrostomy, particularly PEG, before swallowing is too difficult because we are worried about the respiratory function. So we are talking about a problem and an intervention before the person feels they need it. And as I said, I've said when we did a session on dysphagia yesterday, feeding is so basic for all of us. And I have a dietitian who says, are you someone who lives to eat? Or are you someone who eats to live? The lives to eat person, which is my partner, who spends a long time cooking the meal and wants to savor it. Swallowing is really important. Taste is really important. For me, who eats to live and has finished the meal in 2 minutes and is accused of not even chewing the food, let alone enjoying the food, it's different. So we need to respond to where patients are. And as someone deteriorates, there are ethical issues of whether we continue with feeding when someone is obviously dying. Do we continue with water and hydration? Or are we withdrawing treatment? It often is a slow reduction and a change to water, but it allows medication. But many ethical issues, and I'll come back to the issues of withdrawal shortly. We've talked about cognitive change, and there are increasing issues. If we are testing people more and more, how do we discuss that? How do you say to someone, we have evidence that you have cognitive change? How does that person cope with that? But it probably is very important that their family know that there are going to be issues, or there are issues, because they can alter what they do. And we can alter what we do in the way of decision making. Because I think there are issues for the patient. How will they make decisions? How will they decide on risks? I have one lady at home who would start the cooking and then leave the house. It didn't burn down, but it wasn't far off many times. But it was also difficult for her family because they didn't know how they could leave her by herself because she was trying to go and drive the car. She was trying to do all the activities which she couldn't actually do. So there's real issues for family of coping. The husband would say to her, we're going to the shops later, and she would put her coat on ready. And we had to help him make the decisions slowly and only include her when it was appropriate. But there are also language issues and communication issues that families need to know about and will often talk to us about. And for us as professionals, how do we assess symptoms if someone has cognitive change? How do we look at maintaining safety? How do we help them make decisions when perhaps there are difficulties in that? How are we with the communication? And I think as us, as part of multidisciplinary teams, other members of the team may have very different views on that cognition. And people may not be able to see the differences and the difficulties. And that can cause real stresses within multidisciplinary teams and real ethical issues. And then when we come to non-invasive ventilation and ventilatory support, we're talking about that much more. We know that it will improve quality of life, increase length of life for many patients, and improve symptoms. So it's a good treatment. And we can talk about those positives. We can say to patients how the symptoms will improve. They will have more energy, they may live longer, they will sleep better. But do we, within our discussions, and ethically, we need to talk about this disease will still continue to progress. Many patients will have issues with the mask. Many patients can't cope with NIV. And we need to think about planning. What's going to happen with deterioration? Do we talk about advanced care planning? That is making a plan while you have the capacity to do so and are able to about what your wishes for the future would be. And then when you can't make those decisions yourself, your wishes are known. Or passing that to a proxy. Now, I know that will vary within countries, and different countries. In the UK, we can do both. I know that varies across countries and legalities. But is it discussed? Often not. And why not? Because of our concerns. This was a gentleman with non-invasive ventilation who I saw at home. His symptoms improved. He complained of nocturia. He wasn't going to sleep. He was waking all the way through the night, and being a good Englishman, when we wake up in the night, we go and pass urine. So he was talking about having problems with nocturia, and the first time he used his non-invasive ventilation, he slept right through the night. I cured his prostate problem. But this is several months later when he's saying, I now cannot speak properly, I now am having problems swallowing, my mobility is poor, I wish I wasn't using this machine. And do we talk about the future? In the NICE guidance we said before, if possible, certainly when starting non-invasive ventilation, the future must be discussed. But there is a reluctance between everyone. I think patients don't want to, families don't want to, we as professionals don't want to, and society varies in its talking about death and dying. And they are complex decisions. At the intervention, we can talk about the options. That information is not easy to impart, and it can be quite daunting, but it is the opportunity. But the day after the non-invasive ventilation started, the patient is feeling so much better. They may be over-optimistic, and where they were thinking about the future and what was going to happen in the future 2 days before, they feel so well now, they're very optimistic, and the options are not discussed. So we need to think of that, and ethically it's really important to maintain more the autonomy of patients to allow them to have those discussions. Because if we don't, how do we talk about it? If we don't talk about deterioration, what happens? Because there is likely to be deterioration in breathing, in general deterioration in cognition. Have we got plans of what will happen? I don't know about your patients, but all changes happen at 2 o'clock on a Sunday morning when no one is around who knows the patient. And if we don't have those discussions, problems happen. But we need to talk about what will happen. Are patient and family aware? Are other professionals aware? Are other teams aware? This was otherwise, we may have patients with tracheostomy who didn't want it. Their breathing deteriorates at 2 o'clock on a Sunday morning. They're in the emergency room, tracheostomy, they're on a ventilator, and then people start thinking, what are they doing? This gentleman actually went into hospital with breathing problems, and he went in for a knee operation and came out with a tracheostomy and a diagnosis of ALS. He made very clear what his views were. He insisted I took his glass of beer, which is in the right-hand corner of the photo, because that was his quality of life. He wanted to show that he had good quality of life as long as he could have his beer. But he made it very clear, with an advanced decision, that he did not want to go back to a hospital ever again. He was fed up with hospitals. If he deteriorated, he knew he was going to die. He wanted to be kept comfortable at home and die at home. And that made the care much easier. He did deteriorate one morning and died at home very peacefully. So it is important to have those discussions before there's problems. Because we know with long-term tracheostomy, there is increased dependency. One study showed that 21% never left the home. Long-term care is needed. There's increased stresses on families. 47% of family members saying their own health had suffered as a result of the person having a tracheostomy. And there is that risk of being locked in. No one's quite sure of that risk, but certainly from the Japanese information, at 5 years on long-term tracheostomy, many patients have restricted communication. And it's often as an emergency situation, as I say, in an emergency department without clear guidance, with respiratory failure. But again, there is evidence that people hadn't talked about tracheostomy in advance because no one had discussed these issues. Many thought it was temporary. And one study, 58% of the patients said they felt pressurized to have a tracheostomy. These were patients who'd said, I do not want this, I never want it, but then at the decision point were really pushed into having it. And that brings us to withdrawal of treatment, whether tracheostomy or non-invasive ventilation. People will start to say they want to have it withdrawn with real ethical issues. People are dependent, they're disabled, their communication is difficult, their cognitive change, and they're asking. And in the UK, and I know again this doesn't apply for every country, in the UK and many countries, if the patient says, I want to stop a treatment, we would have problems if we didn't stop that treatment. So it's a real issue. And if someone is not on 24-hour dependent NIV, they can stop using it, but they may become breathless, and we need to have medication to preempt that. If they are dependent, they will die quite soon, even on non-invasive ventilation, but that might be hours or even a day or two. But they might have severe breathlessness, so we have to preempt that. And that's where the ethics become really difficult. We've done several studies now of the effects of families, of patients and professionals of how they found withdrawal of NIV and tracheostomy. And they knew the ethics. They knew the ethical reasons were fine. This bit knew really well how the ethics were. This bit thought, I'm pressing a button and patient dies. And their heart was saying, you killed the patient. Their head was saying, this is ethically removing a treatment that person does not want. I am doing the right thing. But their hearts were saying other things. And palliative care doctors who had a lot of other experience in ethical decision-making were putting this as a stressful, at 6, 7, 8 out of 10, as stressful decision-making. And it really caused problems within teams, again, because people's understanding of ethics may vary according to their discipline. And obviously you're looking at other staff and volunteers, and families may have different views. So it's a real ethical discussion and real problems. Because we will need to preempt with medication to reduce that risk of breathlessness. And that may mean morphine and midazolam, and maybe an infusion. And if people are interested, the Association of Palliative Medicine in the UK has produced guidelines on this, giving ideas of medication and ways that can be involved for all professionals to use, and they're available on their website. And then finally, end of life. We're talking in many different ways. This is all the different ways that people in the UK talk about dying without saying the word dying. Is it the same in other countries? Do you all have the euphemisms, the things that happen that people say? I spend a lot of my time in the hospice saying people die. We must talk about dying, not passing over, not passing away, not going to a better place. We must talk about dying. But there are real conflicts within families. Families may have very different views. All our families are different. All our families have issues. We are all very different. But there are often fears about talking about death and dying. There's that magical thinking, if I talk about dying, it's going to happen. There might be the existing family issues. The disputes and arguments that have happened for years come to the fore when someone is dying. And do we as professionals give varying and inconsistent messages? Do we talk about becoming iller, which probably for us means they're likely to die soon, but for other people may not seem quite as bad? And it may be talking about morphine. This was an area I found when I first was involved with MND and ALS, that neurologists didn't want to talk about morphine. That's changed dramatically in the last 30 years. But we have evidence. This was a study from many years ago where we looked at the amount of morphine and how patients receive morphine. The mean duration of use was 95 days. So patients were starting morphine on Monday and dying on Wednesday, but it wasn't that Wednesday, it was 3 months later. And because people were worried about the end-of-life issues, we looked just in the last 3 days of life. And the dose does increase to some extent, but very small amounts over those last 3 days of life. And the doses are still not high. And then the ethics on hastened death. Now that, again, varies so dramatically from country to country. It's in some ways a societal decisions and societal things that are made. That there are some countries where euthanasia or assisted suicide is very common, particularly for patients with ALS, and others where we cannot, like the UK. But I still get requests for hastened death. And I think we have an ethical directive to listen to those requests and get behind those requests to why someone might be asking. Is it let me die? I don't want to be kept alive. Is it I want to die because I'm so low, my symptoms aren't controlled, I'm depressed, I've got so many problems which we can address? And sometimes it's kill me. I want autonomously to make that decision to die. In countries where we can't hasten death, we have to cope with that. We have to listen and try and see if we can help people see the quality of life they do have and make the most of that. Because I think we do need to find out why patients are asking. What is bringing that to the fore? And often, like the lady who was told not to look on the internet because you're going to choke to death, she was terrified of choking to death. And there are still books and websites and articles that talk about choking to death. In the UK, we have a particular problem because every time legislation is suggested for assisted dying, someone with ALS is brought out and talks about not wanting to choke to death. And we spend the next 6 months talking to all our patients that that is not an issue. There are studies now of well over 500 or 600 patients from palliative care situations, and one person choked to death in those studies, and that was in 1968 at St. Christopher's, one of the first patients they looked after. So we can say that distressing death is no more likely with ALS than with any other disease. But we need to listen to those fears. We need to think of how families are hearing those requests, and how are professionals? How do we cope with our stresses? I'm sorry, other people may have heard me say this before, but in the UK, doctors have no problems with stress. We have the highest suicide rate, divorce rate, alcohol rate and drug abuse rates, but we have no problems with stress. Nurses tend not to have problems with stress, they say, but I don't know about outside our hospitals because you can't smoke within the hospital. Every morning before the shift, there are groups of nurses getting as much nicotine into their system as they can possibly get before they go on duty. Do we talk about the stresses for us as professionals, as us as teams, as us supporting each other? And with the European Association of Palliative Care and the European Academy of Neurology, we came up with consensus document on palliative care in neurology in 2016. And one of the important areas was education for neurologists and palliative care doctors, but also support for us as professionals. So, we need to listen to patients and families. We need to help them talk about the future. We need to think ahead. We need to help them plan for the best, but consider the worst. And that's quite difficult because we try not to think about the future. All of us do that. We don't want to think about the future. We have our plans of what I'll do when I'm 95, not knowing it may not happen. Thank you very much.
Thank you very much. We have a lot of time for questions. Perhaps Wolfgang will ask the first question.
Well, thank you very much, David, and I'm so glad that palliative care is now included in neuromuscular disease. I note a certain enthusiasm for guidelines, and the concern is, are we trying to modify the patient's disease according to the guidelines? I saw this very much in the Kübler-Ross stages of death, when in our nursing school, patients are abnormal if they don't behave according to these guidelines. So how do we tackle that?
I think we always have to think of guidelines. They are a guide. It is not compulsory to go down the path in a certain way. And I will deal with illness differently to other people. My family is different to other families. And it's the individuality of the disease. We were hearing earlier the difference in phenotype. But there is an even bigger difference in our emotions, our emotional reactions, our ways of coping. And I think that's the challenge, to find out where that patient, that family is, and how we can help them. And there will be some patients who will not accept any of what we're saying, will never die. They're now going to keep alive forever. They are going to be the next Stephen Hawking. You know, I've only been ill for 2 years, so I've got 48 years left. And we have to help them be a little bit more realistic. And there'll be some people who will be very negative, who perhaps can be encouraged to be a bit more positive. And I think that's the real challenge. The guidelines are there, but they guide us, and it's the individual we have to respond to. And the ethics, it is, you know, we talk about autonomy and the ability to allow able patients to make the decisions they want. We have the challenge if someone has cognitive change with that. But we have the challenge to do the other aspects of ethical debate. Doing good, beneficence, not doing harm with non-maleficence, and also thinking of the justice, helping people have the rights and responsibilities, and to think of wider issues of society. And that's a bigger ethical problem. And I think something that medical, social, political systems don't actually think about very often, of how we look at things for the whole community.
Thank you. I think, you talked about the fear of professionals, and I think it's analogous to kill the messenger, who brings bad news to the king, and has nothing to do with the content of the message, but gets killed for bringing the news. And I've found that more recently it's easier because a lot of patients or their families have already looked on the internet And they come in and they have an idea of what their diagnosis is. But in earlier years of my practice, when people really didn't know, it was extremely difficult to give this news because we believe in truthfulness, but there didn't seem to be a good way. There was no good way to bring this really horrible news. I don't know if you have comments.
It is going to be horrible news, however it's given. The evidence from some of the studies are time was important. The doctor who spent 3 minutes telling the diagnosis was seen very differently to the doctor who spent 30 minutes. So it's being seen to be having time and being seen to have empathy. That was the biggest thing that patients could spot, that their doctor was empathetic. And actually, I think patients and families understood this was a horrible thing for the doctor. And they were wanting to nearly work with the doctor to make this as easy as possible and make it work well. And I think there are more and more studies saying this is a process. And there is a tendency, I know, in the UK, it's difficult for a neurologist who may only have that appointment, and a follow-up appointment can't be very quickly after that. But, it may be this is the first part of telling you the diagnosis. We'll talk about it again in a week or 2 weeks. And what we said in our NICE guidance was one of the team could follow up very quickly within 2 weeks, so that someone from the multidisciplinary team who knew about ALS would see the person quite quickly afterwards. Because there's evidence in cancer that people don't hear anything after the word cancer. And I'm sure it may be the same. Someone hears ALS and that's the end of their communication. They've not heard whatever you say after that. So it needs to be said again and again. And I think with gastrostomy, I think, again, it's a process of decision-making. It's not a sudden thing. So it's giving information and building on that over a period of time. I hope that gives some ideas, but it's going to be difficult. And it's something about helping us as professionals to cope with that as well. And having the opportunity to talk about how difficult it is sometimes.
Leonard noticed that it's important to assess mental capacity and to make decisions. So in this supportive care, is cognitive impairment some kind of disqualification of the patient?
No. But I think if we are aware there are the issues, and I've had the same as Leonard, when I sort of took 10 patients and said how many I thought might have cognitive change, and I put 1 or 2. Then we did the testing, and it was 6. And I was going, oh, I've missed things. Some of the nurses said, well, actually his wife told you some of those things, but, she certainly told us that there were problems. But I think knowing, we just have to alter how we care for people, just to some extent. So it may be, there is a tendency to say, would you like this treatment, this treatment, that one? 6 treatments, now do you decide? With someone who's got cognitive change, it may be, let's get down to 1 treatment you want or you don't. And involving your family and other members of the team in helping you with that. So it's changing it rather than stopping it. It's changing that decision-making.
I could see, if you've been following the patient for a while and it's not one simple diagnostic appointment, that you might eventually use the word death. But that's a very rough word to use in a patient interview. I can't remember when I've used it. The patient may say to me, how long have I got? Or how long do you think I've got? Or what, But even the patient doesn't want to use these words, and I'm not sure. This is very difficult.
I think the problem is if we use other words which people don't understand. And, you know, the very poorly we find in our hospital is very difficult because nurses and doctors say he's now very poorly. In medical language, that means they've got a day or two to live. In other language, it means you're not going to go to work tomorrow. And I think there is a need to have a way of talking that, yes, you're not going to get over this, there is no cure. We are looking at quality of life, we are trying to help you do the things you want to. That doesn't stop us, you know, that doesn't mean it's negative. It's trying to say, how do we make the most and the positive aspects? But I think there are times when patients, if they're given time, may say, how am I going to die? Because they may have read the things.
I think even, you know, our nurses, when they call a family in the middle of the night and they say, you may want to come into the hospital to see your relative, they don't actually say because this person's going to die almost immediately, but they call you and say, you might want to come in now to see the person. It's
And I think that can be a problem because some families pick up on that cue.
yeah.
And others say, oh no, it's okay, I'll come in the morning. And then they are very angry. But no one said he was going to die tonight. They just said I needed to come. So I think it's getting, and I think it is a society problem of actually talking about death and dying. And we actually have an organisation in the UK called Dignity and Talking About Death, to try and get people to talk about dying and death generally, not just in neurological disease.
Thank you. Are there any other questions? Then thank you very much. Thank you, Claude.
Thank you, Vera.
It was a pleasure.
And this is some kind of wink.